Hello everyone, I am MNM Baptista and I'm part of the Laboratory of Biosystem Dynamics. This laboratory is in the ARFO building and is part of the Faculty of Medicine and Health Technology at Tampere University. Our group focuses on in vivo dynamics and regulatory mechanisms of bacterial gene networks by combining experimental techniques such as RNA-seq, flow cytometry, and time-lapse microscopy with dynamic models. In this video, I'll be talking about a study we did about the sequence and dynamics of E. coli genes with dual sigma factor preference that was published in 2022 in the BBA Gene Regulatory Mechanisms journal. Inside E. coli, there are sigma factors and RNA polymerases, and when these two components bind, it forms the RNAP holoenzyme. This holoenzyme can then recognize the promoter region of a gene with the help of the sigma factor. After promoter recognition, transcription of that gene occurs, forming an RNA. Finally, translation of that RNA occurs and proteins are formed, which go on to participate in various biological processes. So, since RNAP needs sigma factors to express genes, E. coli uses different types of sigma factors to recruit big sets of genes to adapt to different perturbations. For example, cells growing in optimal conditions will mostly have sigma 70. However, when cells begin to experience starvation, sigma 38 will start to be produced as well to help the cell adapt to that environmental perturbation by increasing the expression of genes that have a promoter that is recognized by sigma 38. However, because there are more sigma factors than RNAP core enzymes in the cells, sigma factors compete to bind to an RNAP. Therefore, the shift in levels of sigma 38 factor will also lead to a decrease in gene expression of genes that are dependent on sigma 70. However, there's approximately 5% of genes of E. coli that have promoters that can be recognized by two types of sigma factors, and most of these types of genes have a preference for sigma 70 and for sigma 38. We refer to these genes as sigma 70 plus 38 genes. This rare set of genes spiked our curiosity, not only regarding their role in biological processes, but also because it's not immediately clear how they will behave when the levels of these two types of sigma factors change inside the cell. Does it depend on how similar they are to sigma 70 or to sigma 38 dependent genes? So we went to investigate how they behave when cells transition from exponential to stationary growth. That is, what happens to these genes when the cells that have mainly sigma 70 start also having sigma 38. To investigate this question, we assessed gene expression by flow cytometry and by RNA sequencing in the exponential and stationary phases, as seen in the two vertical lines in the growth curve depicted in A. As we can see from figure B, we found that the sigma 38 levels increase approximately 30 times when cells transition to the stationary phase, but RNAP levels remain approximately the same. We then investigated the genome-wide gene expression levels when cell transition to stationary. Here are the distributions of single gene expression. Let's focus on the mean value of distributions, which are represented by the vertical bars. There is an increase in the expression of sigma 38 genes, as we can see in the vertical blue line, which is approximately 3. Also, we can see a downregulation of the expression of sigma 70 genes. If we look at the average behavior of these genes, shown as the vertical green line, it is slightly below 0. Interestingly, the expression of sigma 70 plus 38 genes was nearly as upregulated as the expression of sigma 38 genes, as the average behavior of these genes, shown as the vertical red line, and is very close to the blue line. To try to explain these gene expression changes, we then investigated sigma 70 plus 38 characteristics and interactions. We found that the promoter sequence of these genes is more conserved when compared to other cohorts of genes. So we ask ourselves, could the sequence be a regulatory mechanism of sigma 70 plus 38 genes? We then compare the promoter sequence with the gene dynamics. To investigate the sequence, we first extracted the consensus sequence from sigma 70 genes and sigma 38 genes. That is, the sequence that are made up of the most frequent nucleotides of sigma 70 or of sigma 38 dependent promoters. Using these consensus sequences, we calculated the p distances d sigma 70 and d sigma 38, which express the fraction of nucleotides from the sequence of sigma 70 plus 38 promoter that differ from the sigma 70 or the sigma 38 consensus sequence. Using the difference between these p distances, we found that the sigma 70 plus 38 genes without input transcription factors are correlated to their promoter sequence. In fact, even the simplest of correlations, that is, a linear correlation, explains very well the relationship within the promoter sequence and dynamics, as we can see with the R² value of 0.9. Therefore, for these genes, gene expression can be predicted from how similar or distinct the promoter sequence is from promoters that are only sigma 70 dependent and from the promoters that are only sigma 38 dependent. From this result, another question comes up. How does the sequence affect changes in gene expression of these genes? To study this, we created a model. And to create this model, we first assessed how gene expression happens for genes that prefer only one sigma factor, that is, sigma 70 genes and sigma 38 genes. In this case, transcription of this gene happens as two independent processes. However, for sigma 70 plus 38 genes, the formation of RNAs can happen in two ways, shown here as the two reactions, either by an RNAP sigma 70 or an RNAP sigma 38 recognizing the promoter. In these reactions, the rates KT sigma 70 and KT sigma 38 control the binding affinity to the holoenzymes and are expected to differ between promoters. Since the number of RNAP sigma 70 and RNAP sigma 38 in the cells are significantly larger than 1, we can simplify the transcription reactors. And we can further simplify the reaction of transcription by merging both these reactions into a single transcription event. To introduce the sequence dependency in the model, the transcription rate constants are set to be sequence dependent, using the p distances d sigma 70 and d sigma 38. That is, the transcription rate made by RNAP sigma 70 is dependent on how similar or different the promoter sequence is to the average sequence of a promoter that is recognized only by sigma 70. And similarly, the transcription rate made by RNAP sigma 38 is dependent on how similar or different the promoter sequence is to the average sequence of a promoter that is recognized only by sigma 38. Now that we set the model, we can investigate how the promoters sequences, that is, the sigma 70 and or the sigma 38, influence the response strength of sigma 70 plus 38 genes to changes in the levels of sigma 38. To do that, we assume that the transcription rates are comprised of basal transcription rates, called here KT0, sigma I, and a sequence-dependent function f to account for the influence of the p distances. Given this and the analytical solution of our model, we found that the changes in RNA expression were best described when we assumed that the p distances affected the rate constants exponentially. The surface on the left, which was created with sigma 70 plus 38 genes with transcription factor inputs, was applied to sigma 70 plus 38 genes without input transcription factors and found that the RNA expression was even better explained by this exponential relation, as we can see by the high R² of 0.98. At this moment, a major impediment for synthetic biology is the inability of predicting the dynamics of genes from their promoter sequence. We know that the promoters control the dynamics, but there are many other factors in addition to their sequences, such as transcription factors, global regulators, and supercoiling. So from the sequence alone, we are unable at the moment to predict their dynamics. When we remove all the other factors that I mentioned, we can predict the dynamics from the sequence. And to prove that, recent papers constructed several synthetic promoters differing only in specific portions of their sequences, and their dynamics are predictable from their sequence. However, this has not been found in nature, and this study reports on a cohort of genes whose response from exponential to stationary phase is predictable from their sequence. These genes have promoters with preference for both sigma 70 and sigma 38 and do not have transcription factors. Our next step should be, like the synthetic promoters, to cause mutations to these promoters and see if the model still holds true. That would allow us from then on to use synthetic promoters built from these natural promoters as building blocks for circuits that aim to behave in certain ways between exponential and stationary growth phases. And that concludes this presentation. For more details about this study, please see the publication. The link is in the description box. Thank you so much for listening.